Good day everyone. I'm Ravendra Sabat from BSIT 1E, and today I will be explaining what biotechnology and cloning are. First, biotechnology is the use of living organisms such as plants, animals, or microorganisms to develop products or processes that benefit humans. It combines biology and technology to solve real-world problems. One great example of biotechnology is the production of human insulin using bacteria. Scientists insert the human gene for insulin into bacteria, and these bacteria start producing real human insulin. This insulin is then used as medicine for people with diabetes. It's a safer, faster, and more effective way to help patients manage their health using science. In simple terms, biotechnology helps improve the quality of life through scientific innovation. Next, cloning is the process of producing an exact genetic copy of a cell, tissue, or organism. This means that the new organism has the same DNA as the original one. A well-known example of cloning is Dolly the sheep. We also have a frog and a rodent. Going back, cloning has many scientific uses, such as helping preserve endangered species, improving livestock, and advancing medical research. In conclusion, biotechnology is the broader field that uses living things for human benefit, while cloning is one of its techniques that allows scientists to create identical copies of organisms. I hope you guys learned something new about biotechnology and how it helps improve human life. Thank you for listening and enjoy the rest of the video.
My name is John Reynan Baladat, 1 in BSIT from the Group 3. The other cloned animal expect from the Dolly the sheep was a frog successfully cloned in 1952 by scientists Robert Briggs and Thomas Kim at the Institute for Cancer Research in Philadelphia, USA. They use a method called nuclear transfer where the nucleus from a frog embryo cell was placed into an egg cell that had its own nucleus removed. This experiment was the first to show that cloning an animal was possible, and it became an important step toward future cloning achievements such as Dolly the sheep in 1996.
Hello everyone, Shakla Level BSIT 1. In 1996, an event changed the world of science and sparked widespread debate. This was the successful cloning of the first animal from an adult cell. Her name, Dolly the sheep. Ang tupa na si Dolly ay klinoon sa Roslin Institute sa Edinburgh, Scotland. Siya ang unang mammals na klinoon mula sa isang adult somatic cell. Dolly's cloning was not just an experiment. It was the first time a mammal had been created from an adult somatic cell, a feat that led to significant scientific and ethical discussion around the world. Who exactly was Dolly? She was a female fender sheep born on July 5, 1996. More than her bred, her existence have a new dimension to human understanding of life. What's so special about her? Dolly was the first mammal clone from an adult cell. This was a major step in science because before her, many doubted that such a feat was possible. Her existence proved that what seems impossible can be made possible. How was she made? Her cloning was performed using a process called somatic cell nuclear transfer or SCNT. In this process, the nucleus of an adult cell is transferred to an egg cell without its own nucleus, a complex procedure that requires extreme precision. And what was the effect of it? Dolly's existence opened up new possibilities in animal cloning, but beyond that, it also sparked widespread debate about ethical, religious, and philosophical issues. The question of playing God was revived. Where can she be seen today? Dolly has passed away. Her body is now on display at the National Museum of Scotland. She remained an important part of their science and technology collection, a permanent reminder of a groundbreaking moment in the history of science.
My name is Mark Aldrin Babilichao, BS81E from Group 3.
Aside from Chip and the Frog, we also have Cheeky Little Rodent, clowned in Hawaii in 1997. Unsurprisingly, Kumulima was the first successfully clowned mouse. She lived until the ripe old age of 2 years and 7 months. This was a great achievement for her creators. She produced 2 litters and was ahem later retired. She lived 2 years and 7 months longer than most mice and had 2 healthy litters of babies, proving clowned animals could reproduce normally. Her success helped scientists clone over 50 more mice, showing the method worked reliably. After her death, her preserved body and the paper with her tiny footprints were sent to the Smithsonian Museum to honor her role in science. Cumulina's story shows how cloning advanced research but also made people talk about the ethics of copying life.
Hello everyone, my name is Christine Prinsana from BSIT-1E. I will tackle about genetically modified organism or GMO. First, what is GMO? GMO are plants, animals, or microorganisms whose genetic material has been altered using genetic engineering techniques. This involves inserting DNA from one organism into another to introduce desirable traits. Here, one example of GMO. As you can see in the left side, it's smaller, while the right side is bigger. Next, how are GMOs made? Selecting a gene. The first step is to identify a gene with a desirable trait. Next, inserting the gene. The gene is inserted into a transfer vector such as a plasmid and then introduced into the host organism. Benefits of GMOs. GMOs offer several key benefits. They can increase crop yields, reduce the need for pesticides, enhance the nutritional value of foods, and even make crops more drought resistant, which is crucial in changing climates.
Clarice Bulgara from BSIT 1E. What is GM crops? GM crops are agriculture plants whose DNA has been altered through genetic engineering to introduce new, desirable traits. These technologies allow for the transfer of specific genes to enhance crops with characteristics like pest or disease resistance, herbicides tolerance, and improved nutritional value, ultimately aiming to increase crops yields and resilience to challenging environmental conditions. What is the purpose of GM crops? The purpose of GM crops is to provide farmers and consumers with beneficial traits such as increased resistance to pests and diseases, tolerance to herbicides, and enhanced nutritional context, leading to higher crops yields, reduced pesticide use, better food quality, and potentially greater food security for a growing global population. What are GM crops? GM crops are plants whose DNA has been changed using modern biotechnology or genetic engineering. Scientists modify their genes to give them special traits that don't naturally occur in the species. Examples of GM crops. Soybeans are made into GM crops through genetic engineering, where a useful gene from another organism is inserted into the soybean's DNA using a bacterium or gene gun. The modified cells are grown into plants and tested to ensure the new traits is effectively stable and safe before being sold to farmers.